history, procedure, and types of fish. So if we look at the fish technology or fluorescence in situ hybridization, then it is used for detection and quantification of nucleic acids in their cellular environment. This technique has a lot of applications in diagnostics, as it can detect disease cells that contain abnormal chromosomal arrangements. So if we look at the principle of fish, then this cytogenic method is actually based on the hybridization of target nucleic acid inside the cell with sequence specific complementary probes tagged with a fluorescent marker. RNA sequences can also be detected using tagged probes. Probes are usually targeted at a DNA in metaphase or interphase of chromosome, and DNA probes tagged with fluorescent marker can be detected with the help of specialized fluorescent microscope. In this diagram, we can see that a probe DNA is making hybridization with the target DNA, and through this hybridization we detect certain types of conditions, chromosomal conditions. So what are these probes actually? So they are complementary to the nucleic acid sequences and tagged with a specific fluorescent dyes, and their size actually ranges from 40 base pair to 1000 base pair. So some of the dyes that are used to tag the probes may include biotin and fluorescein and digoxygenin, etc. So if we look at the history of fish technology, so before 1940 this technology was named as in situ hybridization, but when immunofluorescence based detection methods are developed, they became incorporated with ISH, and now we term this technology as fish technology. So it was Joseph in J. Gall, Mary Lou Pardee and John et al. Who developed fish technique in 1969. So in 1985, Freyburn and Gill first time introduced biotin labeled DNA probe in plant species. So J. Gall, the experiment which was performed by J. Gall and Mary Lou Pardee was actually the experiment in which they tagged ribosomal RNA probes to visualize ribosomal RNA encoding genes in Xenopus laevis oocytes. And it was the first experiment that was the major experiment in the development of fish technology. So if we look at the procedure of fish, then first step is sample that contains the target nucleic acid sequence undergoes pre-treatment in order to facilitate the entry of probe DNA inside the cell. For improving permeability, organic solvents such as formaldehyde are used. Next step is denaturation. In this step, the target nucleic acid sequence and probe DNA is denatured using alkaline or thermal methods so that they can hybridize with each other. So next step is hybridization. When fish probe enters the cell, then hybridization took place. The incubation period is a time when probe and target nucleic acid sequence are allowed to hybridize at a specific temperature. This process may take hours to complete. So hybridization can take place between DNA and DNA, DNA and RNA, while RNA and RNA. So next step is detection and visualization. After hybridization, unpaired fish probes are washed out, and visualization can be made possible through microscope with the aid of many filters, and large number of images are obtained and high magnification is used. So the last step is the analysis, in which the results are further analyzed by counting individual sequence of interest. So this diagram is actually showing the procedure of fish technology, in which they are the probe DNA is labeled with the fluorescent marker, and then the denaturation of both probe DNA and our target DNA occurs, and then they hybridize with each other, and next we analyze through the hybridization with the help of signal that is produced after the hybridization. So if we look at the types of fish, then we have Q fish. It unites fish with artificially synthesized peptide nucleic acid, and computer software is used in order to quantify fluorescence intensity. So telomere length research involves use of this technique. Next type is hybrid fusion fish. In hybrid fusion fish, we uses primary additive emission fusion of fluorophores to produce further spectra through a labeling process termed as dynamic optimal transmission. So about 7 detectable emission spectra are generated using 3 primary fluorophores using DOT. This technique is more faster than traditional fluorescent microscopes. Next step is fiber fish and flow fish. So flow fish is actually uses the flow cytometry to perform fish automatically using per cell fluorescence measurements, while in fiber fish, interface chromosomes are fixed to a slide in such a way that they are extended out in a linear manner rather than being coiled as it happens in conventional fish technology. So this method is achieved by applying mechanical shear along the length of the slide, either to the cells that have fixed to the slide are then lysed, or to the solution of purified DNA. A method known as chromosome combing is increasingly used for this purpose. The expanded conformation of chromosomes helps in higher resolution, and the construction of fiber fish samples is performed routinely in specialized laboratories. So this method is not possible in common or general labs. So this is all from my side. From here, Fatma